Okay, we're going to start today the study of one of the main control systems of the body, that is the nervous system. It's difficult to study one of these control systems. The other big one is the endocrine, without studying the rest of the control systems. Because remember, we divide the human body into systems, chapters, PowerPoints, to organize our study. But all of these things are working together. Okay, when you study the nervous system in anatomy, you see these big divisions: central nervous system, which is the brain, spinal cord, and the peripheral nerves, motor, autonomic, sensory nerves. And you study series of functions, actions of different areas of the brain, functions of the spinal cord, okay, what nerves do, what muscles they innervate, what happens when we have a sympathetic or parasympathetic activation, inhibition. Okay, and that's the good way to study. Understand the details of the little parts, to then later, okay, integrate all that, okay, and try to see it as a single function. Okay, the nervous system for physiologists is a bit different to what you study in anatomy. Okay, we can represent the nervous system as something like this. There is a center, there is an area that is what anatomists call the central nervous system, okay, that integrates information from many parts of the body, receives information from skin receptors, from mucous membrane receptors, from joints, from tendons, from the bone, from the gut, from the bladder, from every part of the body. Okay, some of this information travels following peripheral nerves, wires, okay, in the form of action potentials. That is what we call afferent information or sensory information. It's the input to the system. Okay, if you have any difficulty remembering which is afferent and efferent, for me, what works is A for arrive and E for exit. So the afferent is the one that enters, arrives, okay, into the central nervous system. And the efferent is the response, the output, okay, of what the nervous system considers is more appropriate when it integrates all these signals. And this is like, imagine you go to a restaurant that you've never been before. Someone tells you, oh, there is a nice restaurant there in Brickell, I don't know, somewhere. And you get there, oh my goodness, it's difficult to find a parking here. Well, there is a valet park. And you enter, the temperature is very nice, the people look nice people, the music is not too loud, okay, there is a lot of space, I see the food smells very good, okay, sight, ear, smell. When you look at the menu, oh my goodness, $100 per plate, no, let's go to another place. So we take decisions based on adding up or considering tons of information. Okay, we like weight, pros and cons of everything. The same thing is the brain doing every second. When you are driving, you are not aware of that, but your brain is looking at how many cars, what is the speed of the cars, what cars are changing lanes, what are the traffic conditions, weather, and your brain is calculating your speed, your movements, everything, okay, unconsciously. Now, this is the information that enters, as we were saying, through wires or peripheral nerves, spinal, cranial nerves, optic nerve, okay, all of these cranial nerves that you studied. But then we have other types of afferent information. Okay, for example, the immune system informs the brain. If there is an infection, if there is inflammation, what type of infection? There is a communication between the cells of the skin, mast cells, macrophages, dendritic cells, that send signals that are captured by the brain, trying to understand what's going on. You know, if there is an infection, the brain responds to those signals by increasing the body temperature, okay, the set point in the hypothalamus to allow fighting bacteria. Okay, all these signals of the immune system normally reach the brain following the blood, the lymphatic vessels, okay, moving into the cerebrospinal fluid sometimes. It's a communication that we call humoral. Humor means fluid. Okay, the communication in the body can be cellular, cell-to-cell contact, can be humoral when you release a signal in the fluids. Okay, a more specific one is the endocrine. We release a signal in the blood. Okay, in this case it's a hormone, but it's a type of humoral communication. Then we have the endocrine system releasing hormones into the blood, also telling the nervous system what are the conditions of the body. When we are eating, after we eat something, the pancreas produces insulin. Insulin tells the brain we have enough sugar, so let's stop eating for a while. And that gives us satiety. When we are not eating for a while, we haven't eaten for some hours, no insulin will be present, glucagon is going to increase. Okay, we're going to have other hormones, ghrelin, for example, that will tell the brain, hey, sugar is about to drop. Let's better find something to eat. And if we don't eat, okay, that will signal the liver, hey, start breaking down glycogen, so we can maintain the blood sugar levels. And then we have also something that requires tons and tons of studies, the microbiota. Okay, the bacteria we have in the skin, in the mouth, in the nose, okay, all these bacteria are producing signals, chemicals, that will enter the blood and will be, in some cases, sensed by the brain, and that will alter how the brain functions. You've heard probably about the brain-gut axis, okay, how the microbiota in the gut influences our mood, our behavior, sometimes our health and disease, because depending on how we feel, we are going to improve or not improve when we have a disease. But we not only have microbiota in the gut, also in the skin. So there is also a skin-brain axis, okay, by which the microbiota on the skin, okay, communicates with the cells that we have under the skin, and those cells send signals to the brain for communication. So notice how the afferent branch of the nervous system is not only the peripheral nerves, it's also the endocrine system, the immune system, and for that reason is that we have a specialty that is more than a medical specialty, is a research specialty that is neuroimmunoendocrinology. There are people who study, all the research they do is based on analyzing how the nervous system, immune system, and endocrine system work together, because they're actually one system. Okay, and it's impossible to understand, for example, human behavior or mood or what we call mental illness if we don't understand the responses, okay, to variations in the normal microbiota, or to the presence of inflammation in some parts of the body. Or do you think we are going to behave the same if we have no pain, or if we have knee pain for a week? Our mood is going to change just because of the pain, just because of the being worried about that. Okay, so that's important to understand how these things work together. Now, going to the PowerPoint, we can start trying to understand some basic things about the nervous system. And this presentation, more than nervous system, is called neural control systems. That means we are not going to go in detail to every part of the nervous system to see how it works. That would take 2 years of studying physiology. Okay, we are going to be trying to understand how the organs of the body interact, how they talk to each other, how they understand each other. Okay, we are going to be talking about communication, in this case between neurons. Then we are going to add the endocrine system, we are going to add the immune system to the entire picture. Okay, typically neurons communicate with each other in a language that we call action potentials, so electrical signals from one neuron to another. Okay, and we are going to understand how in our nervous system, brain, spinal cord, we have several circuits, okay, that allow the communication between neurons to perform specific actions. Okay, the circuits are composed of the afferent neuron, okay, that sends signals from the peripheral receptors, that can be external or internal receptors, to interneurons that are present in the spinal cord and in the brain cortex, or maybe in subcortical structures as well. And then we have the efferent neurons that are the ones that send the response to different organs, that are the effectors, the ones that will carry out that response. Okay, you have there these definitions, okay? And there are some times that we perceive the results of the output. Okay? We are nervous, we may perceive the sweaty hands, okay? We may perceive the palpitations. Sometimes we are not aware. I would say in most cases we are not aware, and that is a good thing. It means that we were aware of everything that happens in our nervous system. And trying to understand these, for example, notice this, efferent neurons send action responses, signals to the effectors. And let's say we want to talk to someone. Okay, we want to say a phrase, we think about it. Okay, and then from our brain we have areas, okay, that we call the areas that process language. Okay, Broca's, Wernicke's area. Okay, we are going to organize the phrase that we are going to say. You mean you have a presentation or something, and we are going to send signals to the motor cortex that will send signals to the muscles of the mouth, tongue, etc., so we can speak. So it's not hard to understand. A neuron sending signals to a muscle and the muscle moving. When you want to say a word, or you want to say a sentence, do you know that even when we don't speak out loud, even when we are thinking with words, the tongue, the muscles of the mouth are also moving as if we were talking. Okay, if you want to see how that works, simply think in any phrase that you want, maybe a phrase that contains R's, and then think that phrase in your inner voice several times, listen to your inner voice, and then repeat the phrase while biting your tongue. And feel the difference.
Oh my goodness.
And feel the difference.
I'll say,
So, for our inner voice, for us to listen our inner voice, when you read out loud to yourself, your tongue is moving exactly as if you were talking to someone else. Because to process those words, we are going to stimulate some muscles. Okay, so every thought will have an action, will manifest as a movement of a muscle. Okay, so when we have good thoughts, optimistic, positive thoughts, something good is going to happen in our body. Negative thoughts are going to have also bad effects on the body. Okay, because in this case we are moving the tongue. Also the blood vessels are constricting, dilating, the heart rate is increasing or decreasing. We're not aware of that because we can't bite our heart when we are thinking about something bad. But that happens. Okay, if we could measure our cortisol, our epinephrine, we would see how having bad memories or rumination of bad things will increase the cortisol, will increase the blood pressure, heart rate, and in the long term, not very good results. Now, something important before entering into the study of this is to understand the difference between sensations and perceptions. Sensation is a physiological process that is unconscious. Okay? This has nothing to do with the language that we use every day. This is another language. This is medicine. Okay? When we speak to friends, family, we still say, I'm a very sensitive person. I have a sensation that someone is watching me. But physiologically, that is not accurate. We don't have any sensation. Okay? We have perceptions. Sensation is what happens when you stimulate a receptor. Okay? Something stimulates a receptor, there will be an action potential that will travel to the brain. The brain is going to send signals to other areas. Okay? And we may perceive that or not, consciously. Okay? There are many sensations that are totally unconscious. Or at least we don't pay attention to them. Okay, now perception is the conscious interpretation of those signals. Okay, and the difference is easy to understand. Imagine you're in the movies with your girlfriend, boyfriend, husband, wife, and they touch your hand. Oh, it's nice. Now, even someone that is sitting on the other side that you don't know touches your hand, it's exactly the same. It's hand on hand. No? The stimulus, the sensation is the same. It's the same action potential. In the other hand, but the perception is totally different. Okay, totally different. So that's the best way of understanding the difference. Now, receptors, okay, since we're going to be focusing on the nervous system, okay, we're going to be studying the receptors of the nervous system, then we're going to study endocrine, and we're going to study the immune system. Okay, this definition for them is they are biological transducers. Remember what is a transducer? It's a device that changes one type of energy into another. Okay, we have receptors that will sense temperature, vibration, touch, pressure, chemicals, okay, electromagnetic waves, sound waves, okay, pH of the blood, different receptors, and they will transform that chemical, electromagnetic, or energy, or thermal energy into action potential, which is the language of the nervous system. Okay, it's like you have someone who only understands English, and you're receiving messages from China, Japan, from different countries. You have to translate everything into English. That's what the receptors do. They translate every signal into action potential. Okay, so they convert every form of energy into electrical impulses. And they have specializations. Okay, they take signals from different parts. Okay, they don't respond to every signal. For example, retinal receptors, rods and cones, they typically respond to light and not to other signals, except if that signal is very, very, very strong. Okay, and if you want to prove that is, you simply close your eyes, and without hurting yourselves, you apply pressure on your eyeball. If that pressure is a bit stronger than usual, than physiological pressure, you're going to see like little lights. You may feel pain. So the retinal receptors may respond to pressure as well. But be careful, don't do that because you may hurt yourselves. But if someone pushes your eye very strongly, you're going to see like little lights. So thermoreceptors, for example, we have several types. We have the ones on the skin that monitor external temperature. These are not monitoring the temperature of the body, but the temperature of the room. And then we have the hypothalamic receptors that measure the internal temperature, the blood and cerebrospinal fluid temperature. Some receptors adapt very quickly to the stimuli. For example, if someone puts, or you put something on your shoulder, you put a backpack, immediately you feel that the backpack is there, you can feel every detail. But after a minute or so, you don't even feel that it's there. Okay? Touch, your boyfriend, girlfriend puts your hand there, oh, nice. But in a couple of minutes, you don't even remember that the hand is there unless they remove it. No, those touch receptors, pressure receptors, vibration, they adapt quickly. But the thermoreceptors don't. Okay, try to keep your hand in hot water to see if they adapt or not. Or in cold water. They won't. Because temperature is something that is crucial. That says homeostasis, but I would say for survival. More than homeostasis. Okay. Then we have the nociceptors. Mm-hmm.
So, but where it says may adapt over time, like when you jump into
yeah.
Okay, because when you jump into a cold swimming pool or something, you're like, but after a while you're fine in there, you know? It's like,
yeah, but what is happening there is more over time. Let's say you move to a very cold, you move to Canada. At the beginning, you have very cold every day. Then that is more acclimatation, okay, over there. But that's also true. When you enter in a very cold or very hot water, at the beginning you feel it, then it's like, but that is like numb. This is like a numbness in the body. Okay? But I've done that. This thing that you move from a very hot to a very cold water, but it's uncomfortable. You feel like numbness, but it's very uncomfortable. This is weird. Acclimatation is like when you move to another country with different temperature the entire year. For example, when I came here, I couldn't go to Costco without lots of stuff. Now I go like to the teacher. They are having increased the temperature here. I think that is to save money. No. Acclimatation. We don't feel the same.
So then is the body becoming acclimated to, like in Peter's example, you go to like an ice bath, right? You jump in, you're freezing, and then after a while you can sit there comfortably if you like just kind of meditate or something, right? So what is
but
that?
acclimatation is talking about long term. Long term means years or months or years.
So it's specifically with time.
Because when that happens, there is numbness.
Okay.
You can use an ice pack to make a surgery. Okay. When I came here, I didn't have a health insurance, and I had a little abscess here. And Professor Santos put an ice pack there for a while, and then cut. You don't need anesthesia for that. You numb the area with ice. I didn't need anesthesia. It's a little. It's not gonna go deep. Well, nociceptors, I have something to tell you. Nociceptors are pain receptors. Notice the etymology or the word nosy. Nosy means pain, okay? Pain. And probably you have heard a lot about the placebo effect, right? Do you know what is the antony, what is the contrary of placebo effect?
Hypochondria.
The contrary, what is the contrary there? Placebo is when, let's say you have a medication that you know is not very effective for something, but patient trusts you, medication is not gonna, let's say vitamin C. The patient tells you, I feel bad every morning, I think I have my defenses are low, what can I do? Oh, you could take vitamin C every day. And they, oh my goodness, I feel better, the vitamin C really works. That is placebo effect, okay? That is the placebo effect. Now, imagine the contrary, patient comes, I'm taking vitamin C every day and it's amazing how I'm feeling. Oh, I don't know where you get that from because vitamin C actually doesn't work. And it depends on what type of vitamin C has to be this brand because this is the one that has research on it and the patient starts feeling bad. That has a name. That is called nocebo effect. Okay, nocebo effect. And we are responsible for both of the effects, placebo and nocebo.
Nocebo with an S?
Nocebo, like nosy,
C-E-B-O.
nociceptor, like nosy. And let me tell you, when you hear the word placebo, never think that we are talking about something that is fake. That is how many people use the word placebo. Oh, that's placebo. Like that is water or that is dust. Placebo is simply the activation of the self-healing mechanisms of the body. Okay? 100 years ago, there weren't any antibiotics. There weren't any medication for anything that actually worked. How people got better then? Of course, many people more died of infections and different things, many more than now. But many people would get better. Okay? Why is that? Because we have in the body self-healing mechanisms that are activated if we know how to treat the patient. When I told you the first day, medicine is science and art. That is the art. Okay? Medication is not going to work in the same way if the patients don't trust you, or if the patients are afraid, or they are scared, or they are thinking in the medical bills. It's not going to work in the same way. Okay, so we have to try to put the two things together. So nociceptors can be everywhere: skin, periosteum, the capsules of the liver, the kidneys, the bones, okay, the tendons, internal organs. Okay, and they are going to produce signals that we don't adapt to them. It's not adaptation. Okay, and what we can do is simply modulate how the brain receives the signals. Pain actually doesn't happen in the joints or in the muscles. Pain happens here, in the hypothalamus. Doesn't matter what is going on the skin or in the muscles or the organs, pain is happening here. That's why we can either block the signals. For example, when we use topical anesthetics, what we are blocking is the sodium channels in the nerves. So action potentials can travel. You can even destroy the nerve. When someone has cancer, for example, with metastasis, with a lot of pain, and they don't respond to morphine, we simply inject alcohol in the ganglia, in the nerves, and the alcohol will destroy the nerve. That sounds bad, but what is better? Okay, having an intact nerve or being in pain the entire day, pain that is unbearable. Okay?
Have you heard of pain reprocessing therapy? Pain reprocessing therapy?
There are several types of therapy. Okay?
Because that one is shown like research that like you have like two groups, and the treated group, around 70% of the participants experience no pain after therapy or.
Yeah. There have been several treatments, and what they do is simply they act in the. When we process the pain, there is a neuron that transmits the pain to the hypothalamus, and there is another that we call the gating mechanism, that if we activate that neuron, it will inhibit the pain transmission. Okay, that works more centrally. That is, for example, what paracetamol does. Okay, blocks the central mechanisms of pain. Paracetamol is not an anti-inflammatory. It's not going to act on inflammation. It's going to act centrally. Okay, decreasing the perception of pain. That is what endorphins do. Okay, and for sure you remember, if you have had any injury, immediately after the injury you may not feel any pain, so you have time to run away. Some seconds, then, oh my goodness. Right? We produce endorphins, which is like morphine, but it's produced by our hypothalamus to block all the transmission of pain, and that's why some placebo treatments work. That is the reason why acupuncture works, stimulating the placebo effect and allowing us to release endorphins in that area. Okay?
So I see that the elderly, they don't feel the pain as like their threshold is like higher. So they can withstand pain more than, like maybe a 30-year-old or 40-year-old. Is it because of the receptors in the hypothalamus, or is it because of the actual nerve?
That can have several explanations, and they have to do with degeneration of the nerve fibers and receptors. In the case of diabetes, more, because in diabetes patients there is more degeneration of nerves. Okay, so that's one of the reasons.
Professor.
And also brain degeneration.
So acupuncture is actually placebo.
No, it's not a placebo. Okay, but works. The word placebo is not bad. It's not that it's fake. Okay, like when we say something is not historical, we don't say it didn't happen. We don't have evidence for it. Placebo effect is activating the self-healing mechanisms of the body. Okay, acupuncture and other treatments, hypnosis, for example, they work in the same way, activating the placebo or self-healing mechanisms of the body. Because hypnosis, acupuncture, they work for insomnia, they work for pain, okay, but they don't work for colon cancer, okay, because we don't have, our self-healing mechanisms are good, but not that good. So it works by doing that, activating the self-healing mechanism. So remember, when you hear placebo, it's not that it's fake. Okay, it means that it can be explained by the medical intervention. Okay, let's say you have two groups of patients with pain. Well, they are going to receive, one of them is going to receive a medication that typically we use for pain, the other is going to receive a red pill. No? And if they have exactly the same level of improvement, then the medication that we are using for pain is actually not doing anything. Because the red pill that has nothing, has sugar, okay, is showing the same results as the pill that we think, okay, relieves the pain. If we see the same result, that medication actually doesn't work. Because sugar, as we know, it doesn't relieve pain. No, so there is no difference. Why? Maybe there is no relieving pain. Well, that doesn't work. If there is a relieving pain that is significant, well, it's activating the placebo effect. Okay, that's more or less how this is done, which I repeat is not a bad thing. I wish I could have everything cured by placebo effect and not have any chemical or any medication. No? It's placebo on, placebo off. We could do that. Now we have the receptors for electromagnetic waves, okay, light receptors in the retina. Rods, cones, sensitive to different wavelengths, colors, etc. Then we have the mechanoreceptors. Notice that we are talking here about exteroreceptors that feel the things from outside. Okay, we have the mechanoreceptors for touch, vibration, pressure, present on skin, mucous membranes. Okay, the adaptation varies. Touch and pressure adapt quickly. The proprioceptors they adapt but slowly. And then we have the chemoreceptors that adapt very quickly. You enter into a room and there is a smell, unless it is really bad, that there is a cadaver for 1 month or something. But it's a normal smell, perfume or something.
Like at the morning.
Unless you're very hungry and the smell is good, that you're going to feel it more and more and more and more intense. And then we have the interoreceptors that sense the conditions inside the body. Okay, we have the baroreceptors for blood pressure, which are a type of mechanoreceptor. Okay, we have the osmoreceptors that measure the osmolality of the blood. We're going to be talking about glucose receptors. We have the receptors for hydrogen ions, chemoreceptors measure the pH. And also we have pain receptors in the, for example, the liver, the kidney capsules, they have pain receptors that will be activated with distension of these organs. Okay, we have in the bone also, okay, periosteum, mechanoreceptors, proprioceptors that sense muscle tension, position. Now this asterisk for glucose, actually we don't have any glucose receptor. Okay, if you analyze the physiology, we don't have a sensor. Let me measure how much glucose is there in the blood. The regulation of the blood sugar level requires a very, very, very complex mechanism, okay, that involves up to 14 glucose transporters located in different organs. There are 4 of them that are the most, the ones that we know better, glucose transporter 1, 2, 3, and 4. And also we have this co-transporter, sodium glucose co-transporter that we have in the gut. Remember we explained yesterday the secondary active transport, glucose, sodium, okay, that's in the gut for absorption and in the kidney for reabsorption of glucose. This is just illustrating the way glucose transporters work. Imagine this is a beta cell in the pancreas, okay? Sugar levels increase. You have there the different steps. Sugar levels in the blood increase. We have a greater concentration of sugar outside the beta cell than inside. Okay, now we have facilitated diffusion. Glucose enters inside the pancreatic beta cell following these glucose transporters. That glucose is going to be used to produce ATP. Okay, inside the cells we have an ATP sensitive potassium channel. Okay, the ATP sensitive potassium channel will close when there is ATP present. Now potassium can't go out. So potassium stays inside the cell. Remember the ATPase is sending potassium in and potassium can't leave the cell. So we have an increase in the positive charges inside the cell. That is called depolarization. The inside of the cell becomes less negative. That depolarization will open calcium channels. Calcium channels following facilitated diffusion will enter the cell and will produce the exocytosis of vesicles with insulin. See how you can see all the mechanisms of transport, or almost all of them, present there. And what is this doing here? We are not talking about nervous system. Remember, the body is only one. Okay, the body is only one. Here we have different concepts. Okay, the depolarization, voltage-gated potassium channels. We have ligand-gated potassium channels that respond to ATP, which is a chemical. Voltage-gated calcium channels, they open when the cell depolarizes. We have exocytosis. We have facilitated diffusion. Okay, we have different things. Now, what happens when sugar levels in the blood go back to normal? These channels will allow a bidirectional transport of glucose. So when sugar levels go to normal and below normal, the sugar that now is in greater concentration inside the cell will start moving up. And the exit of sugar will stop the production of ATP. Those potassium channels are going to open, potassium is going to escape, the cell membrane potential goes back to normal, and those calcium channels close, no more insulin is released.
That's when sugar levels go down or are normal?
No sugar below. Okay. Once you don't have more sugar going into the cell, no more ATP is going to be made. Okay. Remember, when you have equal concentration, there is no net movement of glucose. Okay, so you don't have no more entry of glucose, no more ATP, so everything stops.
So when you're measuring blood glucose level, is it not measuring glucose, or is it measuring insulin?
This what?
That when you're measuring glucose level through blood, what's actually being measured, glucose or insulin?
In the blood test,
Yeah.
glucose. We're talking here about cells. Cells don't have receptors for glucose. Okay. Now, for purposes of exams, this is endocrine.
This is what?
Endocrine system. You know that a body is only one body. If we say we are going to test nervous system, okay, this example of the glucose transport, you don't have to study the glucose transporters for nervous system, but you have to study action potential, okay, ligand-gated ion channels, voltage-gated ion channels, facilitated diffusion, exocytosis, different things that you see there. So in the nervous system, we have different types of cells. Okay, the neurons, the most important, the best known cell, fundamental structural functional unit of the nervous system. We already mentioned there are three types, efferent, okay, interneurons that integrate information, okay, and they communicate with other neurons or with muscles or with glands, endocrine, exocrine glands, okay, by means of neurotransmitters, which are chemicals, okay, that are released, okay, and bind to receptors in other neurons in other places. Every neuron in the brain will have connections or synapses with thousands of other neurons. Okay, if you enter into the field of neurobiology, you're going to see how complicated that can get. Okay, there is a branch of neurobiology that is called connectomics, and they study connections, and they have been able to stain individual neurons to see how many connections each individual neuron has, and they are making a map of the brain and understanding all the connections of all the neurons, different areas with other areas. Okay, and then we have the supporting cells, okay, the glia, neuroglial cells. In the central nervous system, we have the astrocytes, okay, oligodendrocytes, microglia, and the ependymal cells. Astrocytes are supporting cells, okay, that work helping the neurons in the metabolism of neurotransmitters and also cleaning the area around the neurons, removing waste products. Oligodendrocytes make myelin. Okay, these cells have like extensions, and they grab axons of different neurons and wrap them with myelin. Okay, then we have the microglia, which are macrophages. Okay, these are cells like the macrophages that we have in the blood, in the tissues. Okay, the microglia are exactly the same. They are eating phagocytosis of debris, and ependymal cells that participate in the circulation, production of cerebrospinal fluid. In the peripheral nervous system, we have the satellite cells, which are more or less like the astrocytes. and the Schwann cells that make the myelin, okay, in the peripheral nerves. We're going to see an axon, for example. We'll have several of these Schwann cells. In between the Schwann cells, you have little gaps that are called the nodes of Ranvier. Okay, you studied that in anatomy for sure. When you study the structure of a neuron, we have to distinguish between the different parts. Okay, we have the cell body. If you look at the slide, the following slide, you're going to see a diagram showing what is the body or soma of the neuron. Notice that it has several extensions that we call dendrites. We typically represent the dendrites like being a short extension there, like little hands. Actually, the dendrites are extremely long. They are very, 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 very long. Okay, the actual ones in the brain. At least in the brain, they are very long. They have to, remember, they make connections with thousands of other neurons. They have to reach very far away. But these dendrites are the receptor zone of the neuron. Okay? The dendrites have little structures. If you see an actual dendrite, it will look like this. If you look at a microscopic, electronic microscopic picture of a dendrite, you're going to see something that is like this. Something like that. Those are the spines, dendritic spines. And those are the ones who actually make a synapses with another. Okay, this is another neuron here. Okay, synapses occur with these spines. And these things are not something that is like fixed. It's not like the wires in your house. These are things that actually are moving. Okay, if you investigate about that, you're gonna not study what you have to study because that's so interesting that you're gonna get so involved in that. I remember a neurologist that was talking about this, and he was saying, I have a friend who is a cardiologist, and he always says that he couldn't be a neurologist because nothing there moves, and the heart is always moving, and the blood vessels contracting, dilating. And he was showing a video of the dendritic spines moving, and he said, you see, it moves. There is movement in the nervous system. Right? We also have some movement. No? Now, why they move? They are simply looking for connections, looking to connect with someone. Okay, when you are, let's say you are studying physiology, some area of your brain is working, and some dendritic spines are going to go to that part to make connections. Okay? And if you don't study physiology, 2 days later, the connections are going to be lost. Because now it has something else to do. You study physiology frequently, the dendrites know that they don't have to go anywhere else. There is always activity here in the physiology area. So they are going to make strong connections. And then you don't have to think about physiology. It comes natural. Okay? Because every time we have to, oh my goodness, how was that? You have to find the dendrite and make the connection with the one that has the information, to retrieve the information. Once the connection is strong, that comes natural. That's how we learn. And for that, we need to sleep. That strong connection doesn't happen if we are not sleeping.
We have no sleep.
Yes.
Okay? And for example, if you have studied or you practice a musical instrument, you know that you need to practice many times something. Let's do a piece of music, a scale. You have to practice it many times. And then, oh my goodness, I don't get it, I don't get it, I don't get it, I don't get it. You go to sleep, next day let me try again. Oh my goodness, I did it. Oh, that's how it works. Sleeping is important.
So while you're asleep, they're making the connections.
We organize the memories, we put the class. Okay, this is physiology, this is pharmacology, this is clinical medicine. Okay? And well, this...
We should take a nap after this class so the connections strengthen.
or during. There are videos on YouTube. When you go on YouTube, says, I saw one the other day, 8 hours long. learn Spanish while you sleep.
I've done that before though, where I've gone to sleep listening to a video and I wake up and I'm like, how do I remember? I don't even remember listening to it.
The other thing that you can maybe not so deeply sleep and you remember some sentences, some words. So this body is taking information. Imagine that we have thousands of dendrites. Some of them, or the neuron is receiving excitatory information from 500 of them, and inhibitory information from 500. What will the neuron do? 500 plus, 500 minus. What is the output of that neuron? I stay where I am. Now, one of the negative or the inhibitory neurons changes opinion and votes yes. Now we have 499, 501, action potential. That's like the Congress. How many favor, how many against? Okay, the law passes. And that is the action potential. Okay, that will send a signal to maybe another neuron, because this may be making a synapses with another dendrite and be part of the 1,000 neurons that is connected with that. Making the decision, do we eat in this restaurant or is it too expensive? Okay, do we buy this TV or the larger one? Do we apply for a credit card or we save money before buying? Okay, all these decisions are made there. So who makes the sum? Who adds up the excitatory and inhibitory stimuli that the neuron is receiving? This area here. That's on hill. Okay? Maybe here we have an excitatory signal, inhibitory, excitatory, inhibitory. There is a calculator. Okay. Yes. So open sodium channels, action potential starts, reaches very fast the other end, and neurotransmitters are going to be released to do another action. Okay, so in this nervous system we have an input, okay, that is through the dendrites in the soma. We have integration or control center, sums all this information in the axon hillock, and then we have the output, okay, of the neuron, that is the axon, okay, and the axon terminal that will release the neurotransmitters. Now, notice the following. There is nothing, when we have these neurotransmitters released, we don't have any arrow coming back here. So this is not a closed loop. There is input, there is integration, there is output, and this is not homeostatic. This is not a loop that is homeostatic. Okay? That is, to see the homeostasis, we have to see this neuron working together with other neurons. Okay, regulating different things like the body temperature, like the pH of the blood, like the oxygen of the blood. And a single neuron, even though it has all these parts, doesn't inhibit itself, okay, to do all these different actions. Then we have the, let's finish this and go to the break, okay? Because we need more real estate in the brain to go to the action potential. Afferent neurons are the ones that inform the brain, that send information to brain spinal cord about the different aspects of the stimuli, for example, the quality, intensity, location. Okay, so inform the brain of what is the type of stimulus, smell, or taste, or touch, or vibration. Okay, and different stimuli will go to different areas of the cortex. Okay, the sensory cortex will receive sensory information. Okay, parietal lobe will receive visual. Temporal lobe will receive taste, will receive smell. Okay, will receive the auditory. Okay, now we're going to have also information about the magnitude, how loud, how intense it is. Sound, for example, or smell. You put too much perfume today. Okay. Location, where it comes from. Elbow is the shoulder. Okay. We have like a map. If you look at the what we call the homunculus in the brain. You are going to see that in the sensory cortex we have like a map. For example, the legs and feet are located in the inner part, in the medial side of the brain, in between the hemispheres. And then we have the trunk, okay, we have the hand, the arms, we have the face, the tongue, etc. So everything is mapped. The same thing happens with the retina. There is a map of each retina in the parietal cortex. We have a map for different frequencies of sound, high pitches, low pitches, different wave sounds in the auditory cortex. We have a map of different smells, good, bad, etc., tastes. And there is a phenomenon, okay, that we are going to be seeing the application. We're not entering now in that because we want to keep it to the basics. But understanding the phenomenon of lateral inhibition is later important to understand certain factors when you do the physical examination. Let's say this is a neuron, okay, that is receiving information from this area here. Every time you press that area, okay, you are producing a mechanical force that will open channels there and will send impulses, okay, to the brain. This is the cortex, spinal cord, then thalamus, then cortex. But we're making only one. And then we have another here, okay, that will take care of this area, and another here that takes care of this area. Okay, so if I apply pressure here, signal is going to go there. Okay, and it's not going to go here or here. But what if I apply pressure here? The brain may get confused. What's that? Okay, if I can perceive the touch very, very well, like a little point, okay, that's what we call a fine discrimination. Okay, and we don't perceive it too accurately. We call it gross discrimination. Okay, sometimes, for example, in the tongue, in the fingertips, okay, we are supposed to have a very fine discrimination. You have a hair there, oh my goodness, have a hair, no, or something, a little piece of food. Put that in your bag. We're not going to feel anything there. Okay, it has to be a bug walking for us to feel it. Now, how our brain knows that this stimulus is closer to this neuron than to this neuron? Well, since we have the stimulus closer to this, when the action potentials are going out, okay, they are going to also send signals to the neighboring neurons. Okay, we have an excitatory signal going up, and then we are going to have inhibitory signals, okay, from this neuron that will inhibit this and this neuron. So our brain knows that how to localize the stimulus. Okay, that is extremely important for visual processing. Okay, if you want to see a sharp image, okay, light rays are stimulating different areas of the retina, and if that doesn't work, for example, we're going to see a blurry because the brain cortex doesn't know exactly from where the blue and the red and the yellow are coming from. Okay, in the case of a healthy person, we take that for granted because that's how we see everything. But when we have degenerative disorders, like diabetes that destroys nerves, destroys parts of the nervous system, like diseases that produce demyelination, multiple sclerosis, or any injury to the peripheral nerves, all those things are gonna stop working. So we start seeing blurry. Okay, and it's not something that we can fix with glasses, because it's a problem with not in the lens, it's not accommodation, it's not the cornea, it's something that is at the level of the brain. Okay, localizing exactly where the stimulus is coming from. And let's have a break. Okay, let's come back at 9:05. No, sorry, 9:10.
We were reviewing the page forward yesterday. We looked at the subject of artificial sweeteners. Artificial sweeteners.
Oh, that's very interesting. But did you find something or?
No, we didn't look into it. We just wanted to ask you in terms of
person doesn't have